So our first segment we want to go through is research and design. And this I feel like would be a review for most of you guys. So I'm just going to start off with the scientific method. And so many of you guys have probably seen this in your bio classes or even some chemistry classes. But we start off usually with observation and question. And so you see something and you think, oh, this might be something that we can do an experiment on. And then we go into background research. The next step would be deriving and making a hypothesis. And then after that, you would, create your experiment to test this hypothesis. And after the experiment, you would be analyzing your results and, what you got. And from these results, you usually make a conclusion about the experiment that you just did. Now, the reason why it's in a cyclical format is because, generally, I feel like more conclusions lead to more questions because researchers and scientists want to know more about a certain topic. And also, it's helpful for the MCAT because sometimes in the experiments that they give, especially for the biology passages and psychology, sociology passages, they kind of go through, about this part of the experiment where you get a conclusion. So it goes around a circle. And then they often ask questions about, what might be future studies that a researcher in the field would want to do afterwards. And so I think it's good to think of it as a cyclical cycle and figuring out what else potentially researchers could be looking at based on their conclusion to really understand the process. Okay, so the first step is observation and question. And so I gave an example here, plants perform photosynthesis. That would be an observation that a student may make. And one question they might ask is, would different amounts of light cause the rate of photosynthesis to change? And so after having this question, right, usually students would do some research about the topic and look into, the properties of photosynthetic plants, look into results from past experiments that were doing a similar type of observation. And so after doing the background research, the next step would be developing a hypothesis. And a hypothesis is a precise statement used to explain a phenomenon that can be tested through experimentation or observation. It's usually in an if-then statement form, and commonly they usually develop a null hypothesis and an alternative hypothesis. Probably the alternative hypothesis is the one that's like more commonly known, especially when you're younger and growing up. I feel like the null hypothesis was something that you kind of learn as you're learning about significance and statistical significance. So the null hypothesis usually is there, and it predicts no statistical significance between the variables being studied. So for example, with our photosynthetic plants, it could be that different amounts of light don't change the rate of photosynthesis. And that would be like the null hypothesis. And then the alternative hypothesis predicts that there is a statistical significance between the variables being studied. And so in that case, it might be something like increasing the amount of light exposure would lead to a statistical or a significant difference between the rate of photosynthesis of the plant. And so usually when we are doing experiments, we are trying to reject the null hypothesis in order to keep that alternative hypothesis or what we're really kind of looking for. And then after the hypothesis is the experimentation portion. And this is really how we test our hypothesis. And so some major, I guess, terms to think about for the experiment portion. Sample size, which is the amount of, like, so in our example, we're using plants. So it would be like the amount of plants that we're using to test our particular experiment. And generally a larger sample size is better. And then there are also constant variables. And so those are things that stay the same in a controlled experiment. So in our plant example, it might be the amount of water, we're giving the plants, the temperature that the plants are in, all these little things. And then after that are controls. And so controls are usually what we are comparing our experimental processes to. And, popular types of controls that they might test you on during the MCAT is like a positive control and a negative control. So a positive control is like, the way I would describe it is, it's something that is already going to happen based on prior, like, rigorously tested experiments. For a negative control, it's something that that an outcome won't happen at all. So usually with plants, I feel like they like to cover the plant completely. And so the plant would not be able to perform any photosynthesis in the event that, like, no light would be exposed to any of the leaves. And so that's usually what a negative control is in plant experiments. And then for an independent, so the main things that we would be modifying are the independent variable. So that's the variable that is being manipulated or deliberately changed in these type of controlled experiments. And the dependent variable is the variable that is being observed and changes in response to the independent variable. So it's usually what we are measuring. And so I gave this, I put up this diagram here, this graph on the left upper corner. So the independent variable is usually always on the x-axis. So it's what we are changing. And the dependent variable is usually always on the y-axis. So what we are measuring and how it's, affected by these changes on the x-axis. So when you are also looking at graphs on the MCAT that they may give you about experiments, just be aware of this. Like, usually the independent variable is on the x. So if you read an experiment that the researchers are manipulating a certain variable and they give you a graph, that variable manipulation is happening on that x-axis. So now I am going to go also into sampling methods. So I previously mentioned sample size, and so there are actually many different ways to, create this sample that you will be experimenting on. So I guess this is especially useful for the psych-soc section because that's usually where they would be testing sampling methods. But simple random sampling is when each individual is chosen entirely by chance and each member of the population has an equal chance or probability of being selected. Then systematic sampling is when individuals are selected at regular intervals from the sampling frame. And then following that, stratified sampling is when the population is first divided into subgroups or different strata who all share a similar characteristic. So potentially an example for that could be like lower class, middle class, upper class type of stratification that share that similar, in a way, financial status. Next, clustered sampling is when subgroups of the population are used as the sampling unit rather than individuals. And so clusters, they usually do groups of, they just use like different subgroups of a population. So that could be, I feel like several experiments like to do by certain, if we're doing plants, certain species, those would be like the subgroups, and those are used as the whole unit. And then next would be convenience sampling, which is when participants are selected based on availability and willingness to take part in the study. So sometimes you might be doing like a clinical trial, and the patients that you are seeing in the clinic on a regular basis are the ones that are easy or convenient to recruit. And so that's kind of, you sample these participants who are just willing and able to do it. And then quota sampling is when interviewers are given a quota of subjects of a specified type to attempt to recruit. So I put parentheses here, like if they require 10 adult men and 20 adult women to be in a study, then the recruiters are looking exactly for this quota and trying to fulfill it. And then next is judgment or purposive sampling, which relies on the judgment of the researcher when choosing who to ask to participate.